While salmon have a high degree of resilience, increasingly extreme and highly uncertain climate conditions, coupled with unprecedented human activity, threaten their survival.
We understand a little bit about fishing. We understand a little bit about freshwater habitat. What we don't understand is the ocean effect. And we have a working hypothesis, which is the juvenile salmon that grow the fastest in their first weeks in the oceans are the ones that survive the best.
So knowing where are these populations that we're interested, where are they out in the ocean, is a really important first step to understand what factors are affecting them when they're out there.
If you don't know what stocks are present, then you're only answering half the question.
The International Year of the Salmon has identified 5 outcomes and related high-impact projects that will create a burst of new knowledge in cross-border and cross-cultural connections. The most visible project is the International Gulf of Alaska Expedition, a voyage that closed a gap in our understanding of the mechanisms that drive salmon production in the open ocean.
This is the first comprehensive expedition to look at Pacific salmon in the winter in the Gulf of Alaska.
We wanted to understand whether or not first winter or the winter conditions would affect the salmon survival in the open ocean. That was an open question. That was our main hypothesis.
Nobody knows too much about salmon wintering in the northeastern Pacific Ocean. We had some fragmental archival data from 1960s, then from one cruise of Japanese vessel in 2006, but no regular grid survey.
Most of the focus of what we understand about salmon is what we understand either in freshwater or coastally. And all of the different countries have been tracking their salmon in freshwater and coastally, and very few other than the Russians have really gone far out into the open ocean.
The International Year of the Salmon is really a collaborative exercise between many partners, but the two main ones that initiated it are the North Pacific Anadromous Fish Commission and the North Atlantic Salmon Conservation Organization. So we were the vehicle that had connections across our 5 member countries, Canada, the United States, Japan, Korea, and Russia, to be able to accept the money and allow us to charter the Russian vessel.
On February 16, 2019, the Professor Kaganovsky left the port of Vancouver. On board, 21 scientists from Canada, the United States, Japan, Korea, and Russia, all tasked with different experiments they would undertake while in the Gulf of Alaska.
This vessel is unique in the North Pacific, and it does really important trawl research in Russia. They use this vessel to do forecasting of pink and chum returns to Russia. That's how experienced they are, and they do a good job.
Being able to work in real time with other scientists looking at similar questions from around the North Pacific is fabulous. It's very, very exciting.
Знаете, обстановка международная все-таки сложная сейчас. Россия, там Канада, Америка. И поэтому мне кажется, что помимо научных результатов еще важно то, что этот рейс сблизит наши народы.
The Russians on their own had decided that the ship would do 2 weeks of survey off the east coast of Russia, duplicating what we're going to do in the Gulf of Alaska.
We adopted a Russian protocol or Russian sampling protocol and actually approach in the first voyage. It was not the same protocol which, for example, Canadians and Americans would do. It was a protocol would allow us to complete maximum amount of stations in a very limited amount of time.
They have a ton of data that they haven't necessarily published in international journals, so they know a lot that we don't necessarily have access to. So having them on the expedition here really is a huge, huge bonus, because we can basically compare all the data we've accumulated here to the huge data set that they have for the western part of the Pacific.
And to just compare what they were seeing there with what we're seeing here, it's really, I mean, there's a lot of stuff that's really similar, and there's also some real differences.
In Russia, we have a lot of experience to conducting such surveys, and we can share this experience to our colleagues, but they can share their experience for some special analysis like DNA.
Part of the agreement was the data that they collect, they've agreed to share it with other people, with their colleagues.
This is thinking outside the box. This is going to where the salmon are. It's finding a way to be there at a critical time in their life history, and it's getting that first information and saying, okay, what's next?
We will have population-specific DNA, meaning we will estimate the abundance of Fraser River sockeye that are in the Gulf of Alaska.
We wanted to understand how the system looks like, the oceanography. We wanted to see how the prey of the salmon is distributed, and we wanted to see what species of salmon we catch and in which conditions, and how intensively they feed.
When we will conduct this research survey, about 70 stations, we will collect all fish, plankton, microplankton, phytoplankton, all levels of ecosystems.
And now with genetics, I mean, we can tell exactly what river each fish comes from.
How many stations were we going to do as we're heading west before we start heading north?
It was 10 in our plans.
Right now I am cutting tiny pieces of fin clips of coho salmon, and I will add these to this plate and incubate them with a buffer, and that will extract the DNA. To know where fish are from is important because in the ocean, fish from different origin basically mix. And so if you want to do management, you got to know where the fish are from to say, okay, this is an endangered stock, we need to preserve this. This is an abundant stock, we can harvest that. These are some magnetic beads that I'm using to purify the nucleic acid, so all the DNA will stick to those beads, and I have a little rack that has little magnets in here, and those will then attach the DNA to one part of the tube, and I can take all the fluid off and the DNA will stay in the tube.
When fish come back from the sea, they have a particular chemistry about them. We can understand what that chemistry, what it's telling us about conditions that they experienced.
I'm doing zooplankton work mostly, so doing nets and then collecting stable isotopes for the micronecton community from the trawl.
How deep was that sample from?
260 meters to surface.
What do you think's in there?
There's a little bit of phytoplankton, there are some FOCs and decomposed, pelagic decomposed.
From a food web perspective, I follow everything from the physics actually through the chemistry. That's the foundation for the phytoplankton, the primary producers. So it's been tracing the phytoplankton through the zooplankton to the sort of what we call the macroplankton, the bigger plankton that the fish like to eat. The mid-trophic levels like the myctophid lanternfish and squid that the bigger salmon will eat, and then obviously all the way up to the salmon. And if we're lucky, we can get some of the predators that might be feeding on the salmon as well.
И лично для меня данное исследование является, отличной площадкой для обмена опытом в данном направлении.
It seems to be that it has a round eyes, big ones. Look at this. It's very strange, yeah. So on that side it looks not round, another side looks round.
So during the wintertime, we have low light. It's very turbulent, very stormy, so there's a lot of mixing, deep mixing, and the phytoplankton don't get enough light to grow. So there's a lot of nutrients, but they're light limited. So yeah, production is expected to be low. We don't expect to see that many zooplankton.
This has been just a really wonderful experience with how well everyone is working together, and the level of enthusiasm between everybody has just been fantastic.
Most of the people wake up for every set, even if it's not their shift. Everyone is working, and everyone is really grateful to be here.
Well, so far we've been lucky with the weather, extremely lucky with the weather, and I think we're getting incredible and remarkable results.
What we have here is salmon, a Pacific variety. We caught a lot of them today. Our biggest catch yet. Looks like we got chum, at least one pink, one shrimp. At least one coho.
Are you happy that we caught so much chum?
Yes, of course. I'm waiting this big catch.
For the salmon that we did catch, we wanted to sample muscle, liver, and gonad tissues, and that's all to better understand the energy density, how much energy does the fish have given the resources that are available in the Gulf of Alaska. So we wanted to sample both the fish and the food sources.
We have two stations right here. 1 and 2. So we're going to be measuring all the salmon that come aboard up here. And each of the fish are going to get one of these little tags so that we can identify the fish wherever it goes on the ship.
My specialty is hydrobiology. I'm searching salmon diet, trophology. Here we can see mostly digested food. Chinook feeds with fish, and we're trying now to determine what species.
Interestingly, one of the species that we found, coho salmon in particular, was feeding on was a pteropod called Cleopyramidata. It's a kind of a sea butterfly. We collected some fish that were absolutely packed with this pteropod, and the pteropods have a carbonate shell, so they're susceptible to ocean acidification. So potentially, ocean acidification could impact what might be an important food item for them during the winter months.
And so we're going to take those back to the lab and try to analyze them to estimate the energy content of the food and of the salmon, and hopefully that'll help us roughly estimate the carrying capacity. How many salmon can the Gulf of Alaska support?
I've been able to collect some good data so far. Mostly I've been collecting some remote sensing data and some hyperspectral measurements. I'm measuring the reflected light from the ocean. So this is a sensor called HyperSAS, and this sensor have basically it estimates how much light is coming from the sun, and also it measures the reflected light from the ocean. So from this measurement we can characterize what types of optically active constituents in the water.
When we first got out to start the survey grid, we were in extremely warm waters, and there was discussion at that time that we were too far south. We should be starting further north because there would not be salmon in these warm waters that were over 8 degrees.
So it's good to have data where there's no salmon, right?
Yeah.
And to get a gradient. We don't want to miss out on the transition. I think that's really important.
Lo and behold, that's where we caught salmon, and that's where we caught pink salmon. It was the only area we actually saw pink salmon.
With their return back to the mainland, all of the scientists started undergoing more rigorous testing from the samples that were caught out in the North Pacific. With these findings, they hope to create a strong research baseline for future expeditions.
Well, we're pretty fortunate that 6 months after the cruise that we're able to get scientists from 5 countries back into a room. So we spent 2 days here in Victoria at the Pisces meeting to listen to each other and learn. And in most cases, this was the first time these scientists, other than by email, have been able to talk to each other. And in some cases, they hadn't seen the outcome of the research.
We have a baseline that has never been available before. We have observations about species distributions that we cannot explain right now.
We have samples in the lab. We have muscle tissues that we're going to be looking at. We have otoliths that we're going to be looking at. And by comparing these with the juveniles and what returns to the rivers next year and the year after, we'll be able to determine if the hypothesis is correct.
What we've learned is stocks that we thought were restricted to the coast are well out into the high seas, like coho, and some of the stocks, Canadian stocks of sockeye, are showing up 2/3 of the way across the ocean.
The waters we were sampling were only 6 to 12 degrees, so that's still pretty cool for them. And so their feeding activity will definitely be less. But we did find fish that were very healthy, robust, and lots of food in their stomach.
I could not understand at that time why so many salmon from Asia and North America migrate in Gulf of Alaska. I realized that salmon distributed in cool water, whose water temperature is between 5 and 7.5 degrees Celsius, and such condition is huge available in Gulf of Alaska.
I think it's really critical we get back out there and go see, is this normal? Is what we're seeing? Because right now we know that the North Pacific is warmer than average by about 1 degree, especially down at depth. 1 degree doesn't seem like a lot because in terrestrial systems it's not very much, but for an ocean it's huge, 1 degree. And so we need to understand what's going on.
As we look at the data and look at the survey area, but particularly try to understand why the pink salmon catches were so small. The pink salmon catches should have been 10 times larger because pink salmon are that abundant. So even that tells us there's something going on about pink salmon that we didn't know before.
What does this mean that there's very few pink salmon? What does this mean that there's so many coho salmon and coho salmon from Canada?
Gulf of Alaska and vicinity is one of the clearest regions for microplastic pollution. So we have microplastics here, but not so much.
We saw these jellyfish a long ways from shore. It's supposed to be a coastal species. Is that normal? And we really don't know because there's been so little sampling out there.
Nobody expects so many of the coastal species. And the way they were distributed and the amount of food they potentially consumed might indicate that there could be a direct competition with salmon.
We went out there really expecting to look for competition, interactions between pink salmon and sockeye and pink salmon and chum, and amongst all the salmon species, not thinking for a minute that jellyfish were going to be part of the equation. So it came as a huge surprise, and it's really, changed the way we need to think about this North Pacific system.
Our lab is looking at energy density of the different species, and that of course includes chum salmon. And particularly one of the things I was wondering is, are there differences between specific stocks sort of in their energetic content?
The information that we're getting from these surveys gives us a chance to look at how much fat content they have, so fat's really important. And at that level, if we're seeing that they're in good condition, then maybe that winter's pretty good, but we're also just trying to find out if they're in poor condition. And if they're in poor condition and competing out there with a lot of other different species and stocks, is that competition impacting survival?
We tested 75 coho salmon while we were out at sea. For 52 of them, we were able to successfully determine a stock identification. And on that level, we're more than 50% were able to positively assign it to the right stock.
In addition to the genetic stock identification, we have been interested in, what are the pathogens carried? What are the other indications of stress and health? And we expect that the vast majority of the mortality that relates to year class strength occurs during the first 6 months at sea.
Most pathogens seem to be disappearing out of the ocean, so that could mean two things. Either the fish that are infected in their early life stage do not survive and therefore drop out of the population, or that a significant amount of the population is actually able to clear these infections.
And one of the things that was surprising was just how critical the zooplankton were. It is really the zooplankton that's supporting the fish.
The nets we used and the sampling design we used allowed us to sample small zooplankton. The type of zooplankton which you usually do not see in the stomachs of the salmon. And while it represents an index of maybe food availability, it didn't provide us with information where the prey items which we see in the stomachs are distributed. That's how we decided that we probably need to change the nets or change the slightly design how we sample potential prey.
We also got some information on some of the other non-anadromous species and what their biomass is. So it's an important survey, especially under the fact that we're starting another marine heat wave, and that started going on during winter of 2019.
A much broader survey will really allow us to look across the Pacific Ocean and where salmon are located relative to their prey resources, relative to predators, relative to temperature conditions, and how do they behaviorally choose where to be.
These are very, very important species that support jobs. They have economic, cultural importance. They tie us together across the North Pacific, and they're part of this food web, this ecosystem that is changing.
If you have the ability to predict what is happening and even have the ability to predict returns, that would be huge for people whose livelihood depend on that, but also it would give us the information on how to proactively do things that address maybe limiting factors in the distribution and survival of salmon, so we can actually make sure that we have stable populations that do return
at constant levels regularly.
It's really the last phase, the life cycle, that we don't know anything really truly about. And so we have to fill in that box.
And what we have learned is that the methodology, the approaches different countries have to exactly the same question are different. So we were actually learning from each other and working together to do this right, I guess.
Part of the success was the particular surroundings that everybody found themselves in. A vessel where they spoke a language that they couldn't understand, signs that told them to do things that they didn't know how to do, and but eventually and very quickly they all became family.
We are working towards planning a follow-up cruise, and the big prize that we're after is a 5-vessel survey right across the Pan-Pacific, and informed by what we've learned from this. So looking at what did we learn, what are the hypotheses that we're trying to test.
You know, going forward, there's going to be things that we would change if we did a second expedition, but now we know. We had no idea what we were going to catch when we got out there.
You know, we went out there for 1 year, which was definitely important to start somewhere, but we need to put it in context with other years, other environmental conditions, because then, only then, can we really begin to tease apart the different environmental effects and how they may affect productivity in the Gulf of Alaska and the whole North Pacific Ocean.
We really have no idea what's going on out in the high seas, and that's the one area that we really have a poor understanding. So I think it's a relatively small investment given the economic value of salmon fisheries across the North Pacific.
I think it's remarkable that we're working together internationally, and I hope that that continues, because together we're going to get the answers that we need to know to be able to manage these incredibly important fish going forward into the future.